So the differences between bacteria, which is an example of prokaryotic cell, and eukaryotic cell in general. So cell wall, most of the bacteria, they have cell wall in terms of peptidoglycan. We said it's sometimes present. So when do you get cell wall? So when do you get cell wall in eukaryote? Well, for example, plants. Plants, they have cell wall made by cellulose. We talked about this when in the last exam. The carbohydrate is a form of carbohydrate, and then fungi. They also have cell wall. Do you still remember what is it made out of? It is called chitin. Okay, and then for both eukaryotic cell and prokaryotic cell, they all have cell membrane. Cell membrane is like universal to all cells. Nucleus. Well, eukaryotic cell, we have nucleus. For bacteria, we only have nuclei, an imitation of a nucleus. Organelles, we have organelles in eukaryote, and then in here they said no. I would say mostly no. Oh, yeah. My handwriting using the mouse is not good. Mostly no. As I said that bacteria, they have ribosome. Ribosome is a form of an organelles. They put it here. They put ribosome separately than organelles. Well, if they separate like this, then you can quite know. So ribosome is a form of organelles, but they put it separately. So they said yes for both bacteria and eukaryote. And eukaryote, they are structurally complex. As we can see from the previous two slides, they are larger, and they contain more organelles. That's why they are more complex. They have a nucleus. They have a true nucleus, meaning that the nucleus has a nuclear envelope or nuclear membrane. So we call it a nuclear membrane. Or envelope. So a cell membrane is a phospholipid bilayer. It's only one phospholipid bilayer, but nuclear envelope or nuclear membrane is a double layer of phospholipid bilayer. That means it has four. Well, say it's a double membrane. Yeah, it's a double phospholipid bilayer. Possess membrane-bound organelles. Well, possess membrane-bound organelles. So membrane-bound organelles like mitochondria, chloroplast, lysosome, so on and so forth. They have cell wall. Yes, I already told you that plants, they have cell wall made of cellulose. Fungi, they have cell wall made of chitin. Okay, these two tables, it seems daunting. You have the name of the all these organelles. They have the structure. Structure is the composition. This composition, you have the organelles. They should call it organelles. You have to call it structure. You have the function. So it seems daunting. We will spend a lecture to talk about each and every one of these organelles. So if you have time, if you want to get a preview, you are welcome to memorize all of them now. It will give you a good preview, but if you don't want to, that's okay. We'll go through them anyway. So you can see the differences in the plant cell and animal cell. In the animal cell, it looks like this. The plant cell, it has a cell wall. It is very easy to identify. I just want to point out something. Their structure in the plant cell, their structure you can find only in the plant cell but not in the animal cell. For example, we call this central vacuole. You can find, okay, in animal cell you see a lot of vacuole. In plant cell you also see a lot of vacuole. These are small vesicles that can contain metabolic waste or nutrient, all kinds of stuff. But in plant cell, they have a characteristic, a central vacuole that is so big. It's a gigantic one. And then they have cell wall, of course. Cell wall is the one that animal cells do not have. Last but not least, they have chloroplast. Let me see, where's chloroplast? Oh, here, chloroplast. They have chloroplast. So these three things animal cells do not have. But animal cells, they also have something that plant cells do not have. For example, we have lysosome. Plant cells do not have lysosome. And we have centriole and centrosome. Centriole or centrosome, it is also something that plant cells do not have. And then we have I don't see that. Some rough here. Well, I don't see which one now. Okay, so assume that that's the two that animal cell have or the plant cell do not have. I may add some more to the animal cell. So from this picture, I can only see the lysosome and centriole and centrosome. Centriole and centrosome, you may assume is the same thing. I'll tell you the differences later. Okay, size of the living things. Well, when I teach biology, I always said that, okay, size matters, and then all my physiology students, they will just laugh. Yes. So, for animal like human being or whales, okay, we can see it by our eyes. Okay, rose, flower, eggs. Remember eggs, it's the largest cell that you can see. Animal like mouse and ant, those are the things that we can see by our eyes. And then until when? Until we go to maybe other eggs like frog eggs, human eggs, plant cell and stuff, all these. Well, let me use different color to distinguish them. When you go to frog eggs, human eggs, plant cell and stuff, or bacteria, chloroplast, yeah, you can see chloroplast. Then we need to use a microscope. The microscope you have in the lab, they're called light microscope. And then for smaller, like virus, protein, amino, and so on, we have to use something like electron microscope, which is something that we don't have in our school. Bacteria is, the size of a bacteria is about 1 micrometer. Remember in your lab, the second lab, it should be your second lab, it talks about conversion and metric system and conversion. So we introduce you to meter, gram, and liter. So in science, we all use metric system instead of mile, pounds, or gallon. Yeah, we don't use those. Daily life, we use those units, but in science, we use a metric system. We also ask you to convert between, let's say, kilogram to gram, or kilometer into micrometer. So in here, this is the micrometer, micro. We use a Greek letter mu to specify that this is micrometer. Eukaryotic cell is larger, as I told you before. It is approximately 5 to 100 micrometer. Or when the cell grow in size, and then volume also increases. Following surface area. Now we start to talk about surface area. The cell size and surface area. This is important. It's a very important concept, so pay attention to the next few slides. There is a reason why cells they are so small, because we have to allow the material to freely go in and out of the cell. If we have a large cell, let me draw it out. If we have a large cell, we only have this surface area. On the other hand, if we have several small cells, that the total volume add up will be equal to the large cell, then guess what? We greatly increase the surface area. So all these are the surface area for the small cell, so that material can go in and out, or to go to exchange the material really fast and efficient, because we have all this surface area. So when you have small cells with the same volume, so that we have multiple small cells, it increases the exchange of the material. That's the advantage. That's why we have small cells, but many small cells.